Mm, That's fine. You should have a lot of extra lids for those bottles. I saw all the chipped
Yeah.
ones they had the
All
other
right.
day.
So we good. All right, everybody. Welcome back to another live stream here at the Palacios Technology Lab. We have a special guest today. We have Bill Graham here with us. Thanks for joining us.
Absolutely. Thank you for having me.
Yeah, so tomorrow is the cannabis tissue culture masterclass, and we are pretty excited. We're going to have Bill. He's going to be the one teaching the class. So, I mean, just going to have a little bit of conversation. If you guys have any questions, just let us know on the chat. So just talk about what we doing tomorrow at the class.
First day of the Cannabis Tissue Culture Masterclass is going to be Introduction and Fundamentals, uh, lab safety and personal protective equipment, but very quickly we get into making media so everybody can get their hands into a project that will be using the ingredients we got assembled here, mixing together the nutrient salts, the hormones, sugar, our special house preservative, and loading up these tissue culture vessels and getting them in that cooker over there.
Mhm. All right. So, I mean, I'm going to ask you something like, what do you think is important to work? And why do you think tissue culture is important, especially for a plant like cannabis?
Oh, there are really several reasons. We've been able to clone
Hmm.
cannabis forever
Right.
from mother plants, and of course, we can make excellent plants from seeds. But each of them have their limitations. Seeds are all going to be variable, even within the same cross. And even after a mother has been chosen and phenoselected to be cloned, the huge weakness in that system, and this is universal, and growers everywhere are running into the exact same issue, that mother plants get too big and too old in the garden. They pick up a lot of the diseases and things that have been present in that garden, and they pass them directly to the clones that are being cut from them. So tissue culture, in our circumstance, will give us a way of evaluating the plants as cleaned on the surface
Hmm.
just by the nature of cleaning the plants and putting them on this medium and watching them grow for a few weeks, and the opportunity to have outgrown systemic diseases like viroids, fusarium, and pythium that would move inside of the vascular tissues of the plant, but are much weaker or missing completely from the tips of the plant. So we make whole plants from just the tips, and tissue culture lets us do that.
Yeah. So, I mean, and
Was that
what?
the short answer?
Yeah, that's kind of the short answer. So, I mean, it is great. That's why we're here. You have for you to comment as much as you can. So if I have a plant that has a virus, I mean,
Yeah.
what are the kind of the most common virus on cannabis? I mean, you're talking like
Hemp latent
yeah,
viroid,
hmm,
hop latent
yes,
viroid, lettuce chlorosis viroid, and the cucumber mosaic. Leaf curly
yeah,
viroid,
colic
yeah,
virus.
and it doesn't matter. They all fall in the same category. It's just trash DNA, RNA. But regardless, if it's at all possible to be able to take a piece of tissue from the plant easily, plant it in something like this, which is really easy to make,
Mhm.
and keep it isolated and grow a new plant from it, we can, if not all in one pass, at least gradually eliminate viroids from the entire crop. And plus, you guys can't see it from where you are, but I am looking at a wall of tissue culture containers that collectively are capable of producing 12,000 new clones every 3 or 4 weeks. And it's just between there and there.
3 to 4 weeks. Yeah, we have a couple of shaving units. I mean, just stay tuned. Tomorrow we have the class, so we're going to be doing some live streaming as well. So I'm going to be kind of showing kind of the behind the scenes of the class. Um, so tomorrow is the class. So what are all the other topics that we want to see on
Oh,
the
we have
class?
a good time with it. We After the media is made, we're of course going to have to select tissue to go into culture, and that's when we'll bring out our little bushy veg plants, our tissue donors, and we'll go over those. And that's a good exercise for everybody. But we'll take the tips and nodes from those, clean them, and put them into the tissue culture vessels that we made yesterday. But we will go on before the end of the day to take plants from the tissue culture containers that were started weeks ago. So real quick, um, at the end of tomorrow, the students' plants will look like this. These are the
Right.
brand new ones. After a few weeks, they'll look like this. And we have enough of these, thanks to
Yes.
you and thanks to
Yeah.
Carly. This is only about 3 weeks growth. So 3 weeks, they're not huge, but what has happened in here is we know that this has no fungus or anything growing in it that doesn't belong there. It has added several new nodes as it's grown, so from these pieces, we'll get 12 or 15 new plant nodes from here, and those will get cut up smaller and planted on a rooting medium. So this one is for producing new nodes and not roots, and this medium here is for growing roots on those. This is what nurseries
Mhm.
like Conception or Node will do to produce their new plants. These little guys are only in here for about 3 weeks, and then they get moved on to plugs. And that's still only part of day one. We are going to clean and germinate seeds using tissue culture techniques for the purpose of being able to rescue old seeds. So the risk of old seeds is that they often have fungus on them or trapped in their outer skin tissues. And we clean those away, hydrate them in sterile solutions, similar to what we use for the culture, and then germinate that in sterile media.
Yes.
And if there are any problems, this is where tissue culture comes in. We can excise embryos from those germinated
Mhm.
seeds and put them onto tissue culture medium so that any part, hopefully the parts that would generate new meristems, if they are alive, can use the medium as food to grow and give us the best possibility of a new plant.
Yeah, I mean that's what we call like embryo rescue, right?
Embryo
Embryo rescue,
rescue.
because we have quite a few questions actually lately about embryo rescue. So,
All right.
so just kind of thanks for kind of pointing that out. So yeah, that's something I actually very looking in for it
Cool.
because that's something that is not basically that's not much of something that I have learned that
Oh,
much.
you're going to love it.
So yeah, that's something that I'm really looking forward to learn that process
Yeah, we are going to
more.
dissect flowers.
Okay,
We are
yeah.
going to dissect seeds.
Hmm.
We are going to we're going to cut meristems. We're going to take plant cuttings and start with sizes like this at
Yes.
about an inch and a half. And before the end of the day, we're going to have those things down to less than a quarter inch. And we get to find out from the students,
Hmm.
and this is one of your fortes.
Right.
So don't let him be modest. They are going to open the brand new growth of tip growth of a plant till we can find material that isn't even shaped like a leaf yet, the very apical
Hmm.
meristem dome,
Yeah.
and they will cut that off, put it on a medium, and we will attempt to grow those. But we will make a good share of
Mhm.
like 6 and 8 centimeter node and tip cuts, just that little bit of plant, and put that into artificial seeds. So all the
Mhm.
students here are going to be
Yes.
making artificial seeds that are easy to store, easy to carry, and we can put into tissue culture. We are doing multiplication. We are doing rooting. We're
Mhm.
doing artificial seeds. We're dissecting. And when we're done with all of that, on the last day of the class, we have DNA and RNA day.
Mhm.
So it's like, yeah, we've dissected the stuff down to as small as we can see. And from there, we're going to use our chemical tools, our PCR machine, and our our
Thermocycler.
thermocycler,
Yes.
thank you, to run some PCR tests. So we're going to be looking for viroids by PCR, and we're going to be looking at our plant genders by
Mm.
PCR.
The gender screening, yeah,
Yeah.
yeah. So yeah, that's one of my favorite parts to do. One of the most difficult ones is to do the shoot apical meristem. So
Okay.
like the shoot tip. So that's what you're talking about. So that's something that we're really looking into for it. So to get a shoot apical meristem where
Uh-huh.
we can potentially we have plants contaminated with viruses, we can remove those using that technique.
Yeah.
And each student, we have several microscopes, so each student has the opportunity to do the work by itself. So I think that's kind of the greatest opportunity
Yeah.
about this class.
And I noticed that we have some new microscopes in the class this time.
Yeah, we have a new microscopes. Yes, we are very excited. We have our stereo microscopes. So we're going to be working with those.
Oh, stereo, so they rock out.
Yeah, yeah, sure. Yeah, so we had the students. They will have the opportunity to each one will have the opportunity to use the microscopes. We want to guide them
All right.
to have a success.
Oh.
meristem experience.
Well, our TV microscopes
Mm,
were like 50 or 70 times magnification.
magnification. Yeah.
Your stereo
Mouse
microscope,
are
which means
90.
two eyeballs, by the way,
Mouse are
are
those two are 90. The stereo microscope.
90.
Yes.
All right, all
Yeah.
right. We are going to be really detailed.
Yes.
Cool. You got some tools for that? Yeah,
Yeah.
yeah. We're
Yeah.
going to
Of
share
course. Yeah.
videos with
Yeah. When
everybody.
like I said, again, I'll be uh I'll be live on Instagram most likely. So whenever that we're doing all this meristem, I want to be just looking, going around the lab and looking at everybody who's doing the meristem work.
Definitely
So
follow all that.
yeah.
If you're not already following us on
Hmm.
Instagram, make sure you
Hmm.
do. We will have some live broadcasts tomorrow, so tune
Yeah,
in
of course.
to see what's going on over here. And send questions, please, because what's great
Hmm.
is the wonderful thing about the class, and this is our seventh Cannabis Tissue Culture Masterclass. Yep.
So,
As they ask wonderful questions. The class is a product of what we do and what the people who come to the class want to learn. And so ask anything. Often you are the one who are asking the questions that
right.
they thought of and may have forgotten. So please bring it all on. We've got 3 7 to 8 hour periods to do this work, to get all this information out here. It's a brain breaker. But what's wonderful is that the things that we do here, you can find when you reference research papers, you look at textbooks, you read the simple or the difficult books on tissue culture. The most important thing about all this that I can tell anybody who's watching this is that this is doable by anybody.
Hmm.
So it looks like we have a lot of special equipment here because this is a serious lab, but uh, this can all be done with simple tools at home.
Right.
Once you learn the principles, you get bigger, and that's when you want to get a big HEPA filtered hood like this, which they have on the Plant Cell Technology website.
Mhm. Yeah, yeah, of course. Yeah, we have any materials that you need to start your tissue culture lab. You can go to plantcelltechnology.com. You can find the store there. But yeah, I mean that's something great about like the master classes, tissue culture master classes, is that, for example, the cannabis class, which is a little bit more extensive compared to maybe the my class. The my
Yeah.
class is like the
Or my simple
gen
class.
yeah my
Yeah,
like a general tissue culture. So in 3 days, I mean, you get to learn pretty much everything you need to get started. I mean, you start your own business, your own lab.
yeah,
Like again, also for like my class as well. So, but my class is more like a general. This is a little more kind of specialized, especially like the PCR and gender screening. So,
yeah.
that's great. Talking about that, actually, um, we have announced the new online teacher quarter masterclass that's actually coming on March 23rd. Is it 23rd and 24th? I believe so.
And you're sure about that date?
I think it is. So I think it is March 23rd and 24th is going to be an online teacher quarter masterclass that is coming soon. So you were not able to register for the previous class that we did online. You are anywhere in the world, you can sign up. So make sure to check plantcelltechnology.com to check for that. Let's see what else. Do you have we have any questions on?
Yeah, we do.
Okay.
One question is, would like to know more about photoautotropic micropropagation.
All right. Oh,
Is
oh,
that what?
gotcha. They're different.
Photoautotropic is confusing for a lot of people.
Photoautotropic is high CO2. Yeah. Uh, and that's really it. We usually more popular in rooting. So photoautotrophic uh, tissue culture, and we've really just jumped ahead in all this. What is regular tissue culture? Just
Right.
real quick. Regular tissue culture, what you're looking at right here is we're just growing plants on the same nutrient salts that you use in your hydroponic fertilizer. There are special vitamins that we would add that aid growth. That's an earthquake. It's Washington, D.C. They happen all the time. Foggy bottom.
Just give me
Um,
1 second.
but regular tissue culture. Are we on?
Yeah, we are. And it's just
You
like
got.
we are cutting our face off. That's it.
We were attacked by giant spiders. Uh, and so anyway, this is nutrients, special vitamins, sugar, and then we mix it with agar or a gelling agent to make it defy gravity and hold up plants. But that's what makes tissue culture special. End of story. everything here we're doing starts with adding sugar to this so that it can be absorbed directly into the stem tissue right across the surface, right here. Uh, and as long as we're feeding the plant, we can also control the direction of its growth by providing either rooting hormones like you're used to. Your rooting gel hormones uh, are the ones that would be in this mix right here because we want these plants to start growing roots. But the opposite of those is cytokinins, which prevent roots but really encourage lots of branchy, short nodes, new growth up top, which is what is happening in this container here. But sugar is the big deal. Everything clean, all the weird stuff you see over here. Why do we say pressure cookers? Because we're using sugar. We want to keep it clean. And that's also the reason that we can say confidently that if the plants are growing in here and there's no weird stuff, because everything else likes to grow on sugar too, that these things are contamination free. That and um, in selective instances, we are using the house specialty preservative, plant preservative mixture, to kind of help keep those things down. But if we can prevent them for long enough, they don't live anyway because you have to live or die. So what's one we end up with these wonderful clean cultures. And that was coming from the whole statement about photoautotropic.
Polarotropic.
Sugar-free, is that what prevents contamination?
No, if we do it without sugar, it's just hydro, and we don't get any real special growth out of it. So at that point, you're simply depending on what's inside the plant. Photoautotrophic means we're really depending on photosynthesis, and we're forcing up the CO2. If you've got a grow environment with 1,000+ ppm CO2, you are basically photoautotropic, which is hilarious. It's just since we have them on a sugar medium, that's why it gets a different name. But we would put the plants in rooting on the same kind of rack on sugar medium, but very quickly they start growing roots and calluses, and that the leaves and the new growth become more dependent on the gas exchange. So we would use a vented container, drive up the CO2 and drive up the light. So then we start pushing the light levels to 200 micromoles higher, but with temperature controlled because we have greenhouses here. Photoautotropic, push up the CO2 and the light and try to force them to use more photosynthesis than sugar.
Here's a question I have.
And then the long answer.
Yeah.
What's the biggest challenge you see most beginners have when starting with tissue culture? If you want to repeat the question, I'm not mic'd up.
Yeah, that's it.
Yeah. So what is the kind of the question is what is the biggest challenge new growers um, get when starting with tissue culture?
Really is just kind of getting over their own hesitation to get the kit and get started. I mean, unless you live in a tent, and if it was a grow tent, we're set. It is extremely easy. I'm going to stop shaking the table. The ingredients are super easy to make. We can do it in just a juice bottle. Sterilize it in a small pressure cooker, instant pot, even a microwave.
Like, yeah.
I've done it in a fast boil kettle when I was on the spot. And there you have the medium. Next is plants and cleaning, and that's going to be any jar. You can either use Clorox bleach, or we have our special dichlor, which evaporates better. But we just make small plant pieces. You get them into the container quickly and by clean technique, and put them someplace clean to grow. The grow shelves, which are behind you, are just lighted shelves, and we have HEPA filters run to keep the air clean for extra security. So it's really just getting over those barriers that a lot of people think are difficult, and are really not. I'm really the best example of this because
Right.
I travel with that box. Want to show them the box?
Yes. Want to show
I
that
mean, I will show up with this little box right here anywhere on the planet,
has everything there.
right here, and we're doing tissue culture within just a few hours. I carry some prepared media in tubes. I have the little media ingredient kit, which I took out and left over there, and a cup with holes in it and chlorine tablets to clean plants. These are chlorine spray and alcohol. And I've got my scalpels and forceps.
Yeah.
This, I've done tissue culture all over the world in just a matter of hours with just what's in
All
the
of
box.
that and then a plastic tub.
And
The famous plastic tub,
the
right?
famous plastic tote, correct. And if I didn't have the tote, I did it without it, and it still worked. We got all kinds of little tricks for this. It's Obviously, as everybody gets bigger, we can expand, and all this is scalable. So it's funny, brand new users
Mhm.
uh, of any language have started for just a few $100s,
Yeah.
learned all the basics in just a few hours, and then they just keep doing that and have scaled up to like 500 or 1,000 clones a day on
Oh yeah.
really just the big
Mhm.
version of the exact same stuff.
Yeah, I mean, it's some people, especially the kind of some of my students that have
Hmm.
taken my class, they know the story, but maybe some people that maybe new or maybe haven't heard the story. But they the reason I actually learned to use tissue culture was because of the video that you put. What was it like 15 years ago or so?
You saw my video?
Yeah, it was the video with you doing.
It was my dad.
It was with strawberries.
Yeah, yeah,
You were
strawberries.
doing strawberries. So basically, kind of my story is that I like to grow carnivorous plants,
All right, yeah, love carnivorous
and
plants.
they grow so slow, like super, super slow. And
In
they,
nature,
yes, 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 in nature. But so I was like trying to figure out like how can I make them grow faster, and just doing a lot bunch of reading and stuff. And
yeah,
I figured out I found about tissue culture, and then I look it up, and it's like oh this fancy cloning stuff. Like how do I want to do this?
right,
But I still keep looking around and stuff around, and then I found like a blog about kitchen tissue culture,
yeah,
and I also found couple of videos, but they were all like in different languages. Nor in, oh, by the way,
yeah,
but this time I was around 15 years old
right.
in high school. I was 15 years old in high school. I had no
I
money
was
to
only
purchase
17 and a half, so
to purchase any equipment.
yeah,
Um,
we were in French too together.
right, and so I was in high school.
Yes.
I was trying to learn all this stuff, and then I was like, I found the your video about doing the tissue cutter with strawberries, where you basically did everything in the kitchen
Hmm.
and you use this plastic box.
Yeah, yeah,
And then from there, it's like, oh, boy, is that simple? Let me try it out. So back in the day, I couldn't get any materials.
yeah. Okay.
It was something that was mostly for like industries or labs.
Right.
So I use orchid fertilizer, 10-10-10 fertilizer
Oh yeah. Why not?
instead of my media.
It complete. It has
Yeah.
micros and everything. All right.
Well, you use no, you use the like the 10-10-10 fertilizer,
Sure, sure.
regular fertilizer. I use a couple of vitamins from the pharmacy and agar from like the Asian store.
That's funny. That sounds
And
like Carol Stiff.
yeah,
Yeah.
that's like her.
Another pioneer.
Yeah, another pioneer tissue culture, the home tissue culture.
Yes.
So that's how I start my tissue culture lab. And many people, when they take the class, I show them my lab. I have
Yeah.
a picture of my lab. I'll have to post that. We actually, you know what, we want to post that on Instagram.
All right.
Picture of my first lab.
Yeah.
Because it was pretty neat. It was just a plastic box with some holes and some
Oh, you
gloves.
did the holes. Okay.
Yeah, I didn't did the open one. I did the like the
Like Lauren does.
yeah, like the
Yeah.
holes in here,
Yeah.
and that was so difficult, but I got it to work.
Okay.
So my first time, which it was lucky or I was just too skilled,
Yeah.
that I actually got it to work for the first time,
That's
and from
awesome.
there I turned that was just a hobby because I used to be like an engineer, like I wanted to be an engineer,
Mhm.
and then from there I just switched kind of my career to plant science.
Oh, that's
And
awesome. I'm really
now I'm
glad.
here, and now we are kind of teaching classes together, so it's
Yeah.
kind of pretty awesome.
And it's
Yeah,
also a very small world.
yeah, yeah,
I'm super pleased to
yeah,
to know that that is awesome. I mean, that's really what I wanted to do,
yeah, of course.
because it wasn't there,
Mhm.
and everybody I ran into was like, "How do you do this? What do you do?" We're still doing it today,
Mhm.
um, but that is awesome.
Yeah,
Relatively
yeah,
easy,
because
and even in failure, you had success.
oh yeah, of course, yeah, because a lot of people, because of the Plant Cell Technology channel, they giving me like a lot of credit to me
Sure. Uh-huh.
because of like seeing they see that it's possible for anybody to do tissue culture,
Mm.
but it's like I got it from you, and then
You're like
they're
the Ratatouille of
yeah,
tissue culture.
yeah. So it's like then I get credit also. Like Lauren is also putting some pretty great content. So you don't know Lauren Plants in Jars, you
Yeah,
can
we want to.
check
I
it
want to
check
meet
her out.
Lauren.
She makes great videos about tissue culture.
Hmm.
But yeah, I mean, there's more people,
Yeah.
which is great because it's something that it used to be very like secretive,
Yeah.
a lot of secret, and but anybody can do this
Yeah.
stuff. You just need a little bit of space and just it doesn't even take that long. It's very, very fast to do.
Good time to give a shout out to Carol Stiff.
Yes.
She was home tissue culture.
Mhm.
She was plastic boxes, but I did too. Um, and, and microwave sterilizing,
Oh
you
yeah.
know, so every little thing like that. I connected with her. She managed the home tissue,
I know.
the tissue culture group.
Mhm.
There you go.
Yes, I know.
Which was over 1,000 members on
Oh,
Yahoo, and then they dropped it.
well, she managed the one on Facebook, which is over. Oh, I can be
There's
wrong.
one on Facebook?
Oh, there are several on Facebook.
Oh,
I can be wrong, but it's over. It was, I think, over 20,000.
really?
Yeah,
Okay.
yeah.
Well,
I need to
well,
check it
it'll be
out.
20,001 the end of the day. I
Mhm.
need to get signed up.
Yeah.
She's still doing that?
Oh yeah, yeah.
Oh, really?
She's
Okay,
still
God.
on. Yeah,
Well, how about that? All right.
yeah, yeah. All right. So a couple questions.
Yeah, agar or gelatin was the first.
Not gelatin. Yeah, gelatin's wiggly. You know how it jiggles. We don't want that. Auger sets, it doesn't jiggle, but it is soft enough that when you stab these little skinny
Mhm.
plants into it, they go right in with no resistance.
Yeah,
So
and they are very small, so this can be fragile.
we
It
got
can be
teeny,
very
teeny
fragile.
pieces,
Mhm.
but that's it. What's funny is, I mean, in a spot, I've used gelatin, and we try to, use it at a higher rate, but it's inconsistent depending on where it comes from. And it was only worth it because we were in a spot had to be done that night, and that's all we had.
Right. That's why you go to plantcelltechnology.com and you get your agar or Gelen gum from
That's
there.
right. And you
Right.
carry a little box like that with you. It
Yeah.
has everything in it.
Mm. Right.
Probably do one more.
Okay. One more question.
Yeah,
About Tween 20 and how it's used.
Twin 20. So a question about Twin 20 and how is it used?
I don't.
You don't.
I like a surfactant, but I just
Mm.
don't bother with tween. Tween's the laboratory stuff.
Mhm.
Remember, a lot of this is built on replicable laboratory research and so on. And they're going to use very specific products like that every lab is supposed to have. That's how that goes. When it comes to doing this where you are, and you're watching this because you're not in a research lab, uh, Dawn dish detergent, a clean dish detergent without weird fragrances and so on like that. Blue Dawn,
Blue Dawn is the one.
that's it. Just a drop or two
Mm.
per liter to break a little surface tension.
Yeah.
Just the same as we would with nutrients. We have some great similar products like I think
Polysolvent 20.
that's right,
Mhm.
polypropylene.
Polysolvent 20. It's also very cheap, so you can get that anywhere. Um, and what we use as surfactant, this is only for sterilization, right? Or
Yeah.
you use it in something else related to tissue culture.
Well, it's funny. I actually use it in my feed solutions too. Foliar feeding.
Foliar
One of
feeding.
the big reasons for us to do it is that tissue culture itself is kind of like the ultimate foliar feeding.
Right.
And so roots do an active transport and usually don't need such things. Sometimes you might have a funky media, grow media, and would use a surfactant for that. In tissue culture, it is because we're doing the exact same thing against the plant surface. The surfaces of these stems right here will have the same glandular and non-glandular trichomes on the surface. It has a waxy cuticle as well, which also repels water. And we just want to kind of help that stuff get through those into the softer woody tissues and ultimately the green cambium meristem, that green growth ring inside of each one of those stems, because that's where that's going. Sugar and nutrients will pass through all of that, get to that cambium, and the cambium will take that and build it into wood. So what's interesting, for instance, between these two,
Yeah.
is that you see little skinny stems here
Yes.
on those. And how are those different than the stems in
They are
these, which are
thicker and growing some callus.
that is wood made from the sugar in that solution right there. And so, tween, polysorbate, Dawn,
Mhm.
the tiniest bit. I The medium we made here, we did not put tween in because it doesn't really matter because
Right.
it's soaking for a while. Anything we would be worrying about trying to get past like cuticle, it softens and will absorb in the hours and days that it's in the medium.
Mm.
So it's already happening.
Yeah. All right. Well, thanks everybody for tune out for the live stream. Tomorrow, we're going to the class starts at 9 a.m. Eastern time. So all the way until the whole day, I'm going to be live streaming every now and then. So make sure to check, especially on Instagram. That's probably going to be where I'm going to be doing the most of the live stream. But yes, if you are interested in learning more about plant tissue culture, make sure to check plantcelltechnology.com. Subscribe to our social yeah, YouTube channel, TikTok, Instagram, X, even Facebook as well. And thanks for Bill for being here. Now, where can people can find you?
Oh, thank you. I am universally microclone.
Microclone.
I've got videos and things posted on microclone on YouTube, Instagram, X, Facebook,
Mhm.
all those things. So it's usually microclone or microclone Bill Graham, microclone 100 occasionally.
Awesome.
But that is my global handle.
Global handle. All right. Well, thank you so much. Goodbye.
Thanks,
Bye.
guys, gals.